So can you start by saying your name, your institution, and your role?
Sure. I'm Clelland. I'm a physician scientist at UCSF, and I clinically see patients with dementia, and my lab works on developing CRISPR therapeutics for patients with neurodegenerative diseases.
Okay, great. And can you describe the SCGE research project you're doing in a couple sentences?
We are working on a gene called C9orf72, which is the leading genetic cause of frontotemporal dementia and ALS. And we are working to develop a CRISPR therapeutic that would be able to remove the C9orf72 repeat expansion from the genome, hopefully treating patients.
Great. What specifically do you work on within this research project?
So I lead the group that is working on the C9orf therapeutic strategy. So we do everything from collecting the patient cells and editing them all the way up to figuring out how we can deliver this along with our colleagues to patients' brains and spinal cords.
And what's your favorite thing about either this research or related research that you're working on?
Research is hard. There's a lot of setbacks, but the best parts about it are working with the talented young folks who come to our labs to try to make the world a better place and help patients, and the patients themselves, who are the main driver of everything that I do in my clinical work and in the research lab.
Great. And is there anything cool or interesting that you found in your research while working on your SCGE project? Or unexpected?
Lots of cool, unexpected, and interesting things. So CRISPR is like the way we use CRISPR is like a pair of scissors that can cut the genome and remove damaged parts of the genome, like mutations. And what has been very surprising is that those scissors can do a very small cut, or they can do a cut that is the equivalent of miles long, and it's still very efficient in the human genome.
What is an advancement that you expect or think could happen in genome editing in the next 10 years?
What I will say is we have figured out using patient cells how we can correct this mutation that will help patients with FTD and ALS. So we essentially know what we need to do in the genome to fix it. What we can't yet do is get this into patients' brains and spinal cords to make it a viable therapy. So the next step in our therapeutic development is to develop the technologies really that allow us to deliver this technology safely and efficiently to patients.
How do you or your team engage with patients or patient advocacy groups within your research?
Patients are the start of everything we do in our lab. So I always say the research starts with the patients because that's where the disease starts. So in addition to seeing patients in clinic, we have patient advocates and gene carriers who visit our lab to talk to students and share their experience with family members who have suffered from genetic disease, as well as their own hopes and fears about the future. And we work with advocacy groups to try to develop therapeutics at the fastest and safest possible rate.
How do you consider health equity in your research, or how do you plan to as your research progresses?
Health equity is a critical part of everything we do, all the way from the folks who come to work in the lab from very diverse backgrounds, to the genomic sequencing that we try to do to broaden the inclusion of sequencing patient populations, all the way to the mutations that we choose to elevate for treatment. Health equity is sort of the core of, I think, everything that many folks here in the SCGE are working on, because what we at heart want to do is help as many patients as we possibly can.
Great. And then just lastly, is there anything else that you would like to say about the SCGE program or your research that you think would be interesting or helpful to people?
So we have started with C9orf72, but we're working on several other mutations that affect people in adulthood and cause neurodegenerative disease. So these are often fatal mutations that run in families that can cause anything from ALS to FTD to Alzheimer's disease, etc. And I think as we learn from these early cases through the SCGE early targets, this will really be something that can accelerate for many patients over time.